part 5, which will be the last part of the bacterial survey. We started out with this new group of bacteria, the actinobacteria gram-positive. They are fungi-like, so they have a lot of characteristics that fungi have. I introduced you to corynebacterium, told you nocardia can cause pneumonia, and that was pretty much all I have to say about that. And that for diphtheria, I told you we would come back and talk about that after we talked about the mycobacteriums. So now we're going to go ahead and do that. We're going to go ahead and talk about diphtheria. Okay, so diphtheria, just like we saw with scarlet fever, diphtheria toxin is produced from the DNA of a virus that infected the bacteria. Okay, so we call it lysogenized bacteria, the bacteria that has a viral DNA in it. And so then that is what produces this toxin. For scarlet fever, recall it made a toxin that caused a hypersensitivity reaction, caused all that redness, all the rash, right? For diphtheria, the toxin is different. So you're colonizing the throat, and the toxin is going to damage and kill the tissue there, and it can completely block off the airway. Otherwise, you'll just have a narrowed airway, and it's still difficult to breathe. We call this dead tissue a pseudomembrane. When you look at it, when you shine a light in the throat and look at the throat, it will look like a grayish color. So it's a dead tissue, and it's also very, very, very sticky, which is why it doesn't just fall off, right? So usually if you had dead skin, it would just flake away, and but this is too sticky to be removed. Eventually the body will remove it, but it takes time. Okay, so the other problem is that if that toxin, while the bacteria are reproducing, that toxin can get into the bloodstream, and then it can damage the heart and kidneys, and that can be fatal as well. So you have to hope that the child survives the pseudomembrane, and you have to hope that the child's heart and kidneys are not damaged. So it's very, very lethal. We say it's a 10% mortality rate, but that's taking into account adults and children. But children 5 and under actually have a 1 in 5 mortality rate, so a much higher mortality rate. So get that vaccine. Make sure your children get the vaccine. Make sure you get the booster. Protect babies, protect children. Okay. DTaP, remember, diphtheria, tetanus, pertussis. Okay. So we say that there's antibiotics and antitoxin available to treat diphtheria, but that would require getting a diagnosis before the pseudomembrane formation, which is unlikely to happen. Okay. Sore throat, fever, congestion, these things are all normal. People are not rushing to the doctor with these symptoms. So, until the formation of the great membrane and it becomes very, very difficult to breathe, the children wouldn't even be typically even seen by a doctor, right? So it's very unlikely that anyone would receive treatment in time. If you get this in your skin in a wound, it can cause an infection in the skin that leads to an ulcer, which is an open sore, and it does not scab over. And of course, that then puts you at risk of more infection, right? You're vulnerable, and it is slow healing. So it's something that, needs to be dealt with carefully, but it's not typically, it's not life-threatening like the respiratory illness. Okay. Before the vaccine, over 200,000 cases every single year of diphtheria. So very, very awful illness that luckily we have a vaccine that prevents this. Okay, so Remember, these are the fungi-like bacteria. So we see here, they're kind of this weird club-shaped. They look like really long rods or clubs, right? Because they're thicker at one end than they are at the other end. Um, yeah, just very, very unique bacteria in this group. Okay, so next totally different group of bacteria called gram-negative non-proteobacteria. And we're going to look at chlamydia, and we'll look at, after chlamydia, we'll look at the spirochetes, examples of spirochetes. Okay, so for the non-proteo gram-negative bacteria, I've selected to focus on chlamydia. Chlamydia is typically caused by Chlamydia trachomatis, and it's typically a sexually transmitted infection, though not always. You can have congenital transmission from mother to child, and that can also cause blindness in the child, and it can also cause UTIs, which typically, again, it's typically sexually transmitted. But if you were to have direct contact, I can't really think of how you would have direct contact without it being sexual transmission. But yeah. All right. So anyways, this is super interesting in that we have two different forms of the bacteria in the case of chlamydia. You have what's called an alimentary body, tiny, and that's the infectious form that's going to go to the next cell and infect the cell. So it goes to the cell, it gets taken into the cell, invagination and formation of a vacuole, and then the vacuole leads to the elementary body to turn into the reticulate body form, which is the larger form. It's the form that undergoes cell division, right? It's the vegetative state. And so then you'll get rapid cell division. And so the reticulate body divides successively, and producing multiple reticulate bodies, right? So you get lots and lots of division. And so this, at this point, is about 48 hours into the life cycle. And then once you've had a bunch of cell division, the reticulate bodies start to convert back into the alimentary bodies, at which point then the vacuole will rupture. The alimentary bodies will be released from the host cell, so the cell itself also ruptures, and then that releases a bunch of the elementary bodies into the environment, and it will then infect all the cells that it lands on, and then this will be repeated over and over again. So this causes a lot of tissue damage, right? Every time it replicates inside of a cell, it then will kill that cell, and it has a it's being amplified, right? So like just one elementary body can lead to dozens of elementary bodies. So that's just the unique example of bacteria. We'll see these kind of more complex life cycles and even more complex life cycles when we get to things like protozoa and helminths. So a little bit more about chlamydia. It is the #1 STI in the United States. It can be spread any mucous membrane, so vaginal, oral, anal. Men and women can spread it to other men and women. And for some reason I was looking at something, and there are a lot of people who think that you're less likely or you're even altogether safe from transmitting diseases if you don't ejaculate, and that's just not true at all. Okay, so any amount of sexual activity can transmit the disease, and any of the diseases. Okay, so no immunity will be formed, and that's true of all of the sexually transmitted infections. You can keep getting them over and over again. You're not able to build immunity against it. And of course, as we talked about before, with women, it can be very serious because it can lead to permanent damage, cause pelvic inflammatory disease, and it can even cause infertility. And most infections are asymptomatic. If you do have symptoms, they're usually non-specific, right? So just some abnormal discharge, maybe some burning upon urination, but nothing that's going to tell you, oh, chlamydia, right? Men can have discharge as well, but it's they don't always, and they may or may not have pain or swelling in one or both of their testicles. So again, symptoms are not. I know when we saw our first example of our STI earlier in this unit, we saw that men almost always had symptoms, and it was the women who just basically didn't have symptoms. Now it's both men and women pretty much don't have symptoms. Also, I mentioned co-infection before. So if you have gonorrhea, you're more likely to have chlamydia. So even though they are different in that gonorrhea, typically 80% of men will have symptoms, and chlamydia pretty asymptomatic, but it's also very common for people to have co-infections of gonorrhea, chlamydia, and HPV. And HPV, human papillomavirus, alone is not really, in and of itself, HPV seems pretty harmless, but the problem is that it actually puts women at a much greater risk of developing cervical cancer. And so we really, because of this, we have finally made a vaccine for HPV. You need to have both doses of the HPV vaccine before you're sexually active in order for it to work, though. It is estimated that by the time you're, 25, that everyone 25 and over has HPV unless they've been vaccinated. It's that widespread. So it's even more widespread than any of the other sexually transmitted infections, right? Okay. I mentioned before, and I just want to repeat, that if you ever test positive for any of the sexually transmitted infections, you do need to tell any and all sexual partners so that they can get checked and get treatment as well. It's the ethical thing to do, and it's also the logical thing to do, because otherwise you have any sexual interaction with them, again, you're just going to get the infection back. But even if you don't plan on ever having sexual contact with them again, you need to let them know so that they can protect themselves and protect any people that they are intimate with in the future. Okay, so spirochetes, main examples, I'm going to talk about Borrelia, and I'm going to talk about Treponema, syphilis. Okay, so Borrelia burgdorferi is the bacteria that causes Lyme disease. However, it's spread through a tick, the deer tick. Okay, so you need to take precautions to prevent getting ticks. Ticks typically take about 24 hours to burrow under the skin. Once they're burrowed under the skin, you will not see them. You will not be able to remove them because you won't even know where they are. Okay, good way to protect yourself is to have thick clothes, like thick jeans, thick socks. Tuck the jeans into the socks so that you are creating a barrier, right? Your skin there's no way for the ticks to get to it. You can then also have some type of boot to go over that as well if you really, you want to be careful, or you don't want people to see your pants tucked into your socks. Boots are a great solution to that. Of course, if there's any chance that your arms are going to be coming into contact with ticks, so typically it's like in the ground, ticks could be on the ground, and then they can be on animals. right? Especially deer. So then you would also need to protect your hands, your arms, if there's any chance of that. And then you want to shower before bed. Showering before bed increases the likelihood that you'll remove any ticks that have gotten onto the skin. And really, a third thing that you should do if you've been in an area with ticks is to just have somebody look on your back of the areas that are hard for you to examine yourself, and to make sure that you don't have any ticks. And if they find a tick, make sure to carefully remove it. And there's YouTube videos that will show you how to remove ticks if you're interested. A lot of times we think, oh, this doesn't concern me, I live in California, but you can get Lyme disease living in California, so you should be careful. Okay, what does it look like if you do get it? I think it's about 50% of people will develop a bullseye rash. And so obviously if you see the bullseye rash, you know right away to go to the doctor for Lyme disease. But if you're one of the people who doesn't develop this rash, then you really don't know. You'll just have this, headache, pain, stiffness in the joints, muscles, fever, fatigue, and you just feel like you can't seem to get better, right? It's lasting, it's prolonged. And so then eventually you go to the doctor saying, I have fever and fatigue for weeks now. I just feel terrible, something's going on. And then they'll just have to run a bunch of tests until they finally figure it out. But if you know that there's even a chance you've been exposed to ticks and you develop anything unusual, then you should let them know that you suspect that it could have been a tick-borne illness like Lyme disease. If Lyme disease is not treated, it can go to the central nervous system and cause other disorders and complications, which can be lifelong. So you really don't want to mess around with Lyme disease. Borrelia recurrentis is a different species, not as severe, but it can cause relapsing fevers. So you keep getting bouts of fever. And it can be tick or lice that spread it, the bacteria. Okay, there's also a lot of other species of Borrelia that we don't have time to talk about, but that pretty much will cause the same symptoms of this relapsing fever. Syphilis, hopefully you're familiar with. Treponema, pallidum, any of the mucosa can be infected, and any break in your skin can also be infected. Direct contact and sexually transmitted, right? So this disease actually has 3 stages: primary, secondary, and latent. The primary stage is a painless canker at the site of infection. We have an example here. It can be anywhere, wherever the infection is. So man or woman, it could be genitalia, it could be throat, like I said, anywhere that you get the infection. And it's actually painless. So it's not always, people don't always know that they have that sore because they don't feel it. And it's not necessarily somewhere that's easy to see, right? So the image that we were shown seems very obvious and easy to detect, but like I said, it could be in places that are not easy to detect and you don't necessarily feel it. Okay, then a couple weeks later, you can enter the secondary stage where it spreads to your skin and you develop a rash, and you can even have some hair loss. Then you go through a period of no symptoms, and that can last years of no symptoms. And then finally you get this reappearance of sores, but now these are internal sores developing on the organs, and that's going to leave permanent damage. It can be fatal, and it can be very, very debilitating. And you can also transfer this from mother to child. So congenital serious neurological damage is the main concern there. Okay, so this image is also a little misleading because it looks like an open sore on an arm. And so this can certainly happen, but it can be happening on internal organs and you don't have any obvious, outward appearance. So internal organs. So remember this diagram here. This is the most helpful diagram. I feel like these are misleading and confusing. Well, this is good to show you what the rash looks like. So that is syphilis. All right, now for another spirochete, leptospirosis is caused by Leptospira interrogans, and this is an intracellular pathogen that you get from either touching infected animals or coming into contact with water that the animals have urinated in. So either direct wound or mucosa contact from the urine of infected animals or water containing the urine of infected animals. Okay. And at first you have symptoms of headache, muscle aches, fever, vomiting, and then you start to feel better, and then the fever returns, and then now you also will have symptoms of jaundice, which is yellowing of the skin and eyes because now you have inflammation of the liver. And then you will also have symptoms of a kidney infection because it has spread to the kidneys and caused pyelonephritis. And of course, you know that a kidney infection can be life-threatening. So can, a liver infection. So these are pretty serious. And we would need to just take a blood sample and do a serological test to diagnose it. And then we can treat it with doxycycline. People who are going to be working with these animals, I believe we do have a vaccine available for people who are at risk of contracting this. So this is an example of pyelonephritis that starts actually from a ingestion or entry through a wound on the skin, and it then travels through the body and eventually gets to the kidneys, right? Most kidney infections, as we mentioned, were from E. coli. It goes from your gut to your urethra, and right, so I kind of said that wrong. So from your gut, you excrete your feces, and then some of that can get into your urethra, work its way all the way up, right? So a unique example here of pyelonephritis that does not start out from the urethra and work its way up. So we're pretty much done with our survey of bacteria, but I do want to mention an example of an archaea bacteria that we utilize and has changed the face of biology. So PCR stands for polymerase chain reaction. It's an amplification technique where you take a few copies of DNA from your sample. This could be an environmental sample. This could be a crime scene swab sample. And you do a simple DNA extraction, and then you do PCR, and then PCR makes millions and millions and millions of copies of that DNA. And now you have enough DNA to actually work with. You can do different types of analyses. You can do sequencing, right? Whereas when you had just the little bit of DNA from your original sample, that's not enough to do anything with. You wouldn't be able to learn anything from just a single sample. So the advantage of PCR is that you don't have to be able to grow it in culture, right? You can amplify it synthetically. Okay, so the reason we're able to even do PCR is because we're able to do this cycle of heat and cooling to allow the different enzymes involved in copying the DNA. And one of the enzymes that we're using is called polymerase, that actually puts the nucleotides in the complementary base pair fashion. And Taq polymerase, named after Thermus aquaticus, because this Thermus aquaticus grows in hot springs, its polymerase is able to function under extremely high heat, right? And so this is all the process of PCR rests upon Taq polymerase, right? If we didn't have Taq polymerase, we wouldn't be able to do PCR. And if we didn't have PCR, we wouldn't know that, for example, in just 1 gram of soil, there's over 10,000 different types of bacteria. Why didn't we know that before? Because we couldn't culture these bacteria in the lab, so we didn't know they existed, right? So why wouldn't you be able to culture something in the lab? It can have special nutrient requirements that we just simply are not aware of, and maybe don't even know how to even provide. It might have complex relationships with other organisms, right? Symbiotic relationships that we would not be able to reproduce in the lab, right? All sorts of reasons. And it's really important to understand what bacteria are in ecosystems to start understanding more about the ecosystem, and also, right, not just learning about environmental microbes, but as I mentioned, PCR is able to be applied to a lot of different scenarios, such as like crime scene investigation, paternity tests, right? So there's all sorts of lab techniques that the first step is to do PCR. In fact, you'll see a few of those in our lab later on in the semester. So that's it. That's the end. Good job. Thanks for sticking with it. Sorry we had to do so many individual videos, and it's gone on and on into the week, but I really tried to provide you with everything that you need for your exam in advance. So hopefully listening to these videos, the rest of this week, filling in a little bit of notes, adding that to what you've already been studying, you should be able to do very well on our exam on Tuesday. All right, office hours tomorrow on Thursday, in the morning for lab and afternoon for lecture. Hope to see you there.